Okay, so good afternoon for this workshop. This workshop is called Neurobiology of the Body Clock in Health and Disease, and it's working? Yeah. Okay, this one is it. Okay. And I would like to say thank you for the organizers for the invitation to make this workshop, and I will try to do my best. This is also a great honor for me to have this opportunity to make this presentation. I will try to do my best to get you enjoy during this 4 hours session. I have never done so long sessions, but we will make 3 short breaks, and the whole presentation will be divided into 4 parts, each about 1 hour. In the first part, I will talk about general features of what body clock is. In the second part, I will present a video about how the body clock works in a human and in the reality, and what is its practical, what is the knowledge which we need to know, or what is the knowledge what we can share for non-specialists as well. And in the last part, in the third and the fourth parts, we will talk about the implication of the body clock in diseases like obesity and metabolic disorder, cancer, and mood disorders. So I'm from Hungary. This is as the beginning of the lecture, I would like to make just a short presentation of why we like to be healthy. Hungary is a small country in the central mid-Europe, which is actually famous of its hot springs, and we have the biggest thermal lake in Europe, which makes it quite a good destination for people from around Europe to enjoy health. The other interesting part is that we have a kind of special climate, a bit more dry and hot, which makes a special climate there, and it's also long, hot summers we have. And the third information I would like to share about our country is a bit cultural part, that you can see this is an island surrounded by big language families, and actually this is the only feature of our culture which is now very distinct from our friends, our neighbors, but as looking at the other historical and cultural heritages, we can find lots of similarities with the neighbors. Hungarians like to live health. Sport is very important in the education of youth people. What you can see here, that Hungarians are not much different looking like the neighboring countries, actually. So it's kind of a cultural mixture we have, but we perform somehow well at Olympic Games. Maybe if you don't know much about our country from football, maybe you know this name. An over was made of Ferenc Puskás, who was the top goal scorer in the 20th century. And my city is a smaller city in southwest Hungary called Pécs, and it's about 700 kilometers away from Prague. And the city itself has a quite rich cultural background, quite similar to the neighboring countries. What is interesting to know that we have a very long history of Christian heritage, and we have an early Christian necropolis there as well. And in the 10th century came our ancestors here and occupied this area, and from there again Christian diocese was established there. And the university I'm coming from is one of the oldest universities, although it was not really working continuously from that time. What is an important part of a cultural aspect is that we had about 200 years rules of Ottoman Turks, which you can see still on the cityscape. We have nice mosques there and minarets. And just like many cities, many big cities in Central Europe, we have a cityscape from the 18th century preserved as a part of Austria-Hungary monarchy, or as the rule of Austrian monarchy. And then finally, based on these many layers, this is a really multicultural city I'm coming from. This is a cityscape, and in the middle you can see a mosque which is actually functioning as a Christian temple since 150 years. So this is just to represent what mixture of cultures we have in our city there. About my background, about my scientific background, our university is not much famous in a scientific, from scientific aspects, but maybe one thing to mention is in neuroendocrinology, all of we know now about the hypophysis and the hypothalamic regulation of the hypophysis has been rooted in neuroanatomical studies conducted in our university in the 1960s. And the researcher there was János Szentagotai, who is the most famous of our university. And now, after this introduction, I would like to start the real workshop with the real topic, which is maybe more which you are more interested in. I will talk about the epidemiological impact of running a healthy clock. So why is this topic important? How we can get funded to run research on this topic? What are the practical implications of that? What are the molecular details which we need to know on how the body clock is working? And what are the experimental animal models which we can use to find out therapeutic strategies or new molecular tools to use this knowledge in disease development. This research area is not a very new one, actually. We have several textbooks available on this topic, so if you're interested, the left one is a very practical one. This is not just for health professionals, so this you can use as a handbook this you can use as a handbook for seminars or teaching materials, actually. The right one is a newer one, which is full with scientific data, so it's more academic. Okay, and there are several more books listed here down. Keywords to use to research, to search for this topic, this scientific part of the topic are here listed: chronobiology, circadian rhythm, biological clocks, melatonin, SCN, clock genes, and chronotherapy. These are the main keywords related to this topic. So in the first part of the presentation, I would like to help you to answer these questions. So what is a biological clock? What is running a healthy clock? Who have problems with running a healthy clock? And what can health professionals do with this knowledge? So to answer the first basic question, what is a biological clock? We can say that it's a biological timing mechanism which is useful to predict recurring events, to anticipate, thereby providing optimal health and advantage in survival longevity. So here's one keyword, to predict, which has caught attention of everyone who was interested in predicting future somehow for several hundreds of years or thousands of years. So this is a question which everyone is somehow interested in. Therefore, we still have nowadays many pseudo-scientific ideas which roots back in this question: how to use the knowledge of the nature to predict future. So actually we have to be very careful with this question, and therefore we need scientific evidences to rule out those theories which have no true scientific background in this question. So, for example, in the Oriental medicine, probably you have heard about the Chinese body clock. There are such very fashionable theories today, which you might have heard about, and this we will also examine during this workshop. But the traditional or Western medicine is always aware of benefiting from the scientific method to observe, test, and model, and reproduce, validate results. And we always have to ignore those ideas which just don't work in experimental tests, but we have to accept those which work. So upon examination of these questions, what kind of biological timing mechanism exists, people were already aware for this question in the ancient times. The first record is actually from Andras, who has described that there are some plants, some trees, which move their leaves during the day, and this movement is a nice periodic. So that we say is the first record of investigating of a daily periodic biological phenomenon. We don't have much record from the Middle Age, although we know that the mechanical clock was invented then, and from that day we enjoy the real time measurements in form of clocks, man-made clocks. But then in the 18th century, as people were aware of observation of the nature, the first attempts were to rigorously describe the nature, and Carolus Linnaeus made a very detailed description of many plants. The taxonomy of him is famous, but what is also famous is this flower clock, which is just simply based on the principle which time of day a specific time of plant can open its leaves and close, and by this we can construct actually a flower clock, and in several cities of Europe you have very nice flower clocks actually. Somehow in the 18th century, French noble people were who first described the interesting rhythmic phenomena. That time it was a very popular hobby to observe the sky during the night, the stars, so the astrology actually, and astronomy. And one of these noblemen in France just accidentally recognized that one of his plants in this observing room closed its leaves during the night and opened again during the day. And he was curious about if some external stimulus is needed, so if the light is it which opens that and closes that or not. And what he made, he just got his plant in his wine cellar where the humidity is constant, the light level is dark, and he observed that this plant still moved its leaves in a daily rhythm, although there was no light-dark cycle. So there was a constant light condition, constant dark condition, and the humidity was also at a constant level. Then other French noblemen made interesting observations about in the same plants that these rhythms of leaf movements of the plants are independent of external cycles. And the next observation was very important, that this daily rhythmicity of leaf movement is not exactly 24 hours. So it's always a little bit longer or shorter if under constant conditions observed. So this is actually a biological proof for that there is an inner mechanism which runs this cyclicity. Now here we see a a name which, of course, everyone who is somehow interested in biology knows, but everyone who is Adventist knows also somehow, because in this point I have to say that in spite of the theories which we know, which are his results of his work, he made actually the first comprehensive description of the nature in terms of animals. So I could compare that to Carolus Linnaeus' observations of plants. This he made in animals, and what he has found during his journey related to plants, that he has seen many species, many plant species who do the very same daily rhythmicity. So this was just a description of phenomena, what he summarized, and he dedicated one book for this actually. So this also highlights that these biological rhythmic phenomena are interesting for any researcher. And in the 1932 was the time, as in Germany, it has been proven that if some bean plants of which the periodicity, the period length was different, if they cross that, then in the second generation, the periodicity, the length of the period time is about the middle between what was the two, what was in the first generation. So the first genetic proof for that the biological rhythms are inherited was done in the 1932. The first experiments on animals were done much later, so in the beginning of the 20th century, and the first rhythms were the body temperature rhythms, which were recorded. Then very interesting description of body temperature rhythms was done a bit later, that the daily active animals and the night active animals have also a daily rhythm in the temperature, but the temperature rhythm followed the activity cycle actually. Then very important discovery was done in the 20s, that also food is an important time cue which can synchronize the rhythmicity of the body. And similar work was done in bees in the same laboratory where von Frisch got his Nobel Prize later about decoding the movements of bees. And even more interesting studies were done on the birds, that birds use an internal timing mechanism, their body clock, for orientation, for migration. The first observation in human were done by Sanctorius about daily rhythms, which all who have done studies in metabolism know this story, that he just recorded how the body weight changed during the day. He recorded the input and outputs of the body actually, and these daily rhythms were the first which have been recorded in a human. Then the first rigorous recording of body temperature rhythm was done in 1866 actually in England. And then the first real proofs that these rhythms of our body are endogenous were done in the 60s in cave studies, where some researchers decided to go down in deep caves and live there for several weeks and months isolated from the environment, and they had just no idea about what the real time is. And then with the rigorous recordings of physiology, they have found out that the inner periodicity of the human is not exactly 24 hours, but about 24.2 or 24.5 hours. So I would like to summarize now the common features of these daily biological rhythms, which we can see everywhere in nature. So these are regularly repeating and therefore predictable events in the life. Actually, there is a very great variability in the forms, in manifestation, in big variety of physiology shows this rhythmicity in lots of species. If we consider the period time, we know lots of types of rhythmicity, but this workshop is more about the daily rhythmicity. The first ideas about the mechanism, what can run this body clock, were that it's simply an answer to the environment signals. So if the light is shut on, then the plants open their leaves, and if there is darkness, if the sun sets, then the leaves close. But now we know, and also some researchers have known that for many years, that this is not true. So we know that there is an internal mechanism which runs these periodicities. So this we know that it works isolated from the environmental signals. Well, but the environment is a very important factor to entrain that, to influence that. So let's see, what are the characteristics? So there is a very stable period time for each rhythmicity. For example, this 24-hour point 2 is a very stable periodicity of the human body's body clock, which is entrained every day to run on an exactly 24 hours periodicity. And this is what I just mentioned. So the inner periodicity has to adapt always to the environment, to the exact 24-hour periodicity. And this means, of course, also a novel attitude in science, that we have to consider these daily events in our physiology under some experimental conditions or under some physiological questions or some clinical settings. And this is what I will talk about during this workshop. And one practical information can be that if measuring only once from a biological object may be sometimes not informative. So sometimes it's very important to see the dynamics, how a certain physiological parameter changes. Chronobiology is actually the term for research in this field on biological timing mechanisms. So we can group the rhythms based on their period time to ultradian rhythms, which are shorter than 1 day, or around 1 day rhythms. We will talk about that more details later. Most important is the body temperature rhythm, the sleep-wakefulness rhythm, and many hormones have their rhythms. And we have also longer periodicities, which are very stable, like the female cycle or seasonal rhythmicity in animals. So what are the main principles how to run a clock in a living organism? So it is actually based on a very simple feedback mechanism. To make a very easily understandable model, if we set the temperature of a room on a thermostat to a certain level, then the air conditioning should need some time to reach that time point, that temperature. But as it reaches that level, then the thermostat will shut off. And then it will need again a little bit more time to cool down, and it needs time to sense for the thermostat that the temperature is cooled down, and then it has to shut on again. And that was a cycle which had two components, an activation and an inhibition, actually. So cyclicity in biology needs actually only these components, an activatory component, an inhibitory component, and what makes it cyclic is a delay in the mechanism. So what means if the first component, the activatory component, starts to switch on the inhibitory component, then at a certain amount of inhibitory components, it will inhibit the activatory component. So it sounds complicated, but maybe with this temperature control model you can understand that. And the delay is the delaying mechanism is what defines the period length of that. So actually if we have a periodicity of seconds, then usually we have some mechanism of ion channels, like in a heartbeat, or if it's about minutes or hours, then it's usually a mechanism of enzyme activities or transport mechanisms. But if it's longer, then usually it's some protein modifications or transcriptional regulations involved as well. So what are the key parameters of biological rhythms? So one is the period time, so one cycle length. The other term is the phase. Phase means the actual level of the physiological parameter within the period, actually. So, for example, this phase is the peak phase, or this is the lowest phase of the rhythm or the rhythmic parameter of physiology. What we know that this period length is very stable, very characteristic for a given physiological process or a species, for example. And that means if we would like to experimentally force that biological oscillator to different period lengths, then it will not work just in a very narrow window of period lengths. For example, if we just increase the periodicity for so for a shorter half-short period length with external stimuli, then the biological cycle will not follow the speed actually, but it will just skip the every second stimulus, and it will still keep its own rhythmicity. So we cannot really entrain these rhythms to any kinds of external repetitions. But we can easily shift the phase of the actual cycle. So with an environmental stimulus, we can force or entrain the cyclicity to change the time point of the peak itself, so when the peak should occur. So at the organismal level, this should work so that we have somewhere in the body or somewhere in an organism there is an oscillator which has to be responsible to maintain this rhythmicity. We have to have input structures, input components which sense the signals of the environment, which tell the clock if it has to adjust to the environmental cycles or not. And then we have the outputs which just drive physiological rhythms of the body, for example. And another important aspect of daily rhythmicity is that the amplitude and the shape of the curve of the daily cycle of a physiological parameter can be used actually for longer time measurements as well. This is very nicely seen in yearly rhythms or seasonal rhythms, which are driven by the daily rhythmic shape of the melatonin secretion, the hormone which is produced always during the darkness, which during the summer has a very sharp peak during the short nights of the summer, and during the day its levels are low. But during winter time we have longer nights, and therefore the peaks are more broad, and that means the total amount of produced melatonin during the day is somewhat higher during the winter time and during the summer, and there are biological sensory mechanisms in our body which sense this total level of melatonin, and this change physiology to summer mode or winter mode, which is very apparent in animals or in some aspects of human physiology as well. Yeah, these are the examples for these daily yearly rhythms. So based on the common rules of circadian rhythmic phenomena, which have been discovered almost everywhere in nature, in living organisms, the term biological clocks have been introduced in the 1960s as researchers from many different areas of biology and from medicine made the first really big scientific gathering, and they summarized these common rules. And only later were those genetical components described which are driving these common rules of daily rhythmicities. Yeah, and the genes themselves, which are responsible for the around 24-hour rhythmicity, have been called clock genes. Of them, the first one was discovered in the early 90s. Then comes the question, where is the clock actually? So it's very nice that we see these physiological rhythms, but do we have a substrate which we can say that is the clock? The discovery of the anatomical localization of the body clock was done in the 1970s, early 1970s. And this is the paper which was published about the discovery of the suprachiasmatic nucleus localized in the anterior hypothalamus, which is responsible for daily rhythmicity. Imagine how was the discovery done? So what do you think? How could be this discovered? It's a very simple idea actually, to make some cuts, some lesions in different parts of the brain. That's the basic of neuroanatomical research actually, which was triggering research in the 50s and the 60s and later in the 70s as well. So some lesions were done in laboratory rodents, and rhythmic parameters of physiology were recorded, and then they have identified a lesion in a given part of the brain which was responsible to diminish these rhythms. So, and that is called suprachiasmatic nucleus. This is a sagittal section of a laboratory mouse brain, where we see here the cerebellum, the brainstem, diencephalon, telencephalon, and here this small part is the optic chiasm, the crossing of the optic nerves, and just above this part you see here a dark area which refers with this staining to that here we have lots of cells in a very small area. It's a very compact part of the brain with lots of small neurons compacted in two nuclei, which are the suprachiasmatic nuclei. This is just a map of the mouse brain with the lots of different nuclei which have been discovered with this neuroanatomical research during the years. This is a frontal or coronal section of the mouse brain, where we see here again the optic chiasm, and just above that you see this very compact paired nucleus. Between them you see the third ventricle, the lateral ventricles here. And if we go a little bit more behind, where the optic chiasm will turn to optic tracts, here above still we have the suprachiasmatic nuclei, and where we just see the optic tracts above them, you see the dorsal part, the very last part of the nucleus itself. The next step in identifying a clock function for this nucleus was to do transplant experiments, which meant they removed, or they made lesions, researchers made lesions of this nucleus, and then they put in the operated animals a suprachiasmatic nucleus of another animal, and they were just checking if the rhythm will be restored or not. These graphs are actually a very typical measures of behavior, of daily rhythmic behavior in mice. We call actigraphs, which means one row is 1 or 2 days. We see here 48 hours or 24 hours, and the dark is shown with the activity time. So, and the white is the sleep time. So in one row we see 2 days, and then the experiment goes on. In this point was the lesion done, the operation done, so destruction of the suprachiasmatic nucleus. We can see that the rhythm completely became arrhythmic. And then here we see a transplant of another suprachiasmatic nucleus, but the host animal, in this case, was a special one. This animal had a mutation which caused its body rhythm run with a shorter periodicity with about 20 hours. So that meant that if the suprachiasmatic nucleus of the host was transplanted in the lesioned animal, then we see from the first view still irregularity. But on the right side, right panel, if the same data were plotted against 24-hour days, like here, so if one row is 40 hours long, then we can see that after several days or some week, the synchrony has been established to the new periodicity. So this was the first proof that the body clock is localized in a distinct anatomical localization, which is able to drive daily rhythmicity of physiology, even in the host. The next step was to record this rhythmicity, so how these neurons run the body clock. And with novel methods, it could be done so that fluorescent reporters in transcriptional reporters have been introduced to mice, to the brain, to the mice, which could be analyzed in brain slices in vitro, and then the emission of light of these neurons could be monitored throughout many days. And a video about that I would like to show. I hope the video will work. So this is a several days recording of the suprachiasmatic nucleus. So this is the right and left side, the third ventricle. So you see that the cells switch on and switch off with a daily periodicity. So this was a very elegant demonstration how these cells work. Then the different cell types of this nucleus have been characterized, which meant this very small, tiny nucleus, which consists of about 10,000 neurons in mice, consists of heterogeneous groups. So here in this picture, actually, we see the two major parts of the nucleus. There is a ventrolateral and the dorsomedial part of the nucleus, which have different functions, different neuropeptides they produce. The ventrolateral is more the recipient part, which gets input from the retina, the light input from the retina, from the eye. And the other part, the dorsal medial part, which is more responsible for the rhythmicity, for the coherent synchronization of the oscillating neurons. And that gives the outputs to the other brain parts to drive the daily rhythm in physiology. And the other experiment to prove the nature of this oscillation of these neurons was to inhibit the neuronal activity of these neurons, so the electric activity of these neurons, with a toxin which is widely used in neuroscience, tetrodotoxin. And there, with the infusion of these toxins, we can see that the amplitude of the oscillation diminishes. But after washing out the toxin, the oscillation starts again with large amplitude. And then that means the oscillation actually does not need neuronal connections with each other, or neuronal transmission to each other, or electric connections of the neurons. But how neurons can be synchronized to each other? It has been shown in genetically modified mice that there is a neurotransmitter called VIP, vasoactive intestinal peptide, which is produced by these neurons, and these peptides signal to each other what time it is actually. So in a mice, which were deficient of this gene to produce this peptide become very irregular after placing them under constant dark conditions. If we check the left panel, the wild-type animals, if transferring animals to constant dark conditions, they should always keep their own body rhythm, so that means the rhythmicity is still there, the motor activity period is always occurring on the same time, even though it's constant darkness. But these knockout animals lost actually synchrony of between the neurons, among the neurons, and therefore it was not possible that the rhythm of physiology could, so rhythm in behavior could not be recorded anymore. So this means the neurons communicate. They must communicate with each other through paracrine way, so not just neuronal way. Yeah, and and how this VIP signals the signal coherence to the neighboring neurons, and how it will be put out in electric synchrony is modeled here and on this scheme that actually VIP signals to transcriptional modifications which act back on ion channels, which alter in a daily rhythm the electric features or the activatory features of the membrane potential of the neurons. And finally, what is to know about about the biology of the biological clocks is that these clocks are actually cellular clocks. So all of the components of a clock is localized in one cell. So single neurons have been demonstrated to produce this daily rhythmicity. But to run a behavioral rhythmicity, these neurons must be synchronized to each other, which they do very well. It has been shown that not only neurons, but actually most of our cell types of our body have this molecular clock mechanism, which means that any cell type which we culture are capable to show transcriptional oscillations or cellular oscillations with a near 24-hour periodicity. However, these cell types cannot synchronize to each other well, so they need some external output, which is in the body the outputs of the suprachiasmatic nucleus in form of hormonal and neural synchronizing signals. And we know for some decades that even bacteria possess clock mechanisms which are actually vital for them to time their when to multiply, when to multiply, actually, when to grow, how to time their metabolic activities. So what now we know about the model of the molecular clock is that it constitutes of positive and negative components which are called clock genes, and they work in a feedback fashion, and these genes are different between the different species, although in vertebrates it seems that these are the most important clock genes. In other animals, these are completely different types of proteins. So these are, although well conserved among vertebrates, but plants, fungi, and bacteria use different proteins, but the mechanism how these proteins work is the same principle, based on the same principle. And this picture is just a very annoying picture, but lots of molecular. So this is not for education purposes of non-specialists, of course, but this is just to show how many molecules are involved in the transcriptional level oscillations. And what is interesting that by now we know genetical data that if one single component has a mutation, to what kind of disease it could be linked to. And this is a profiling of mood spectrum disorders or psychiatric disorders. We can see that for most of the components there is some kind of connection which have been described. So although the quality of the picture is not the best, but maybe you can see for that this protein, this kinase is a target for in lithium therapy, for example, in depression, or some single nucleotide polymorphisms have been identified in relation to some psychiatric conditions. So to sum up, we know that the rhythms are inherited, and the circadian rhythms are driven by biological clocks, and we have genetical mutations. We know that these molecular clocks are running in single cells, in different types of cells, were distinct types of cells. And from the 90s, and especially from the 2000s, in the last decade, they just started to map how these components interact. And in parallel, it has evolved another scientific area, chronomedicine, which deals with the timing of treatments and their impacts on health. And now, as you have a picture about biological clocks now based on the results of research of the previous decades, we can have a look at the pseudo-scientific ideas which are related to this topic. How many of you have heard about the Chinese body clock? Please, hands up. Not many, but maybe you will hear about that. So therefore, this is an idea which is a part of traditional Chinese medicine, that throughout the 24 hours, in each dedicated 2 hours, there is a peak of one organ function, in a peak of a function of one organ. So, which means, for example, they say that the gallbladder works between 11 p.m. and 1 a.m. And I just want to highlight one more. The pericardium would work between 7 p.m. and 9 p.m. So, based on these two examples, we can see that we don't know anything related to the function of pericardium. Why should it have a peak of function exactly at that time? So based on what you have heard of the Western research, Western medicine, and the Western type of science, we always have to have a mechanism behind the phenomena which we observe. Of course, this is, as most of pseudosciences must have something which is true in that, and in this case is the observations of human physiology, which may be true for the heart. We know that the heart is activated the most in the morning hours, in the before noon, or we know that maybe the large intestine has more activity in the morning hours. Most of people have a daily rhythm in defecation. So these simply observations made them to make a theory, but we don't have any more biological relevant mechanisms to describe these these phenomena. And unfortunately, I don't know what is this organ. So this is, maybe who know the Chinese medicine knows better. I don't know. Oh, really? Okay. So this is just a picture about that what we know with of Western type of science, that this rhythmicity is a cellular-based rhythmicity of any organ, which must be synchronized, and these rhythms, this rhythmicity is at the molecular level, which results in physiological outputs of rhythms of each organs or each tissues. And this can be measured at different physiological parameters, and the daily amplitude of these rhythms can reach the dimension which can affect outcomes of clinical conditions. For example, the incidence of myocardial infarction or stroke, which is the highest at the time of sympathetic activation, which we have usually in the morning hours. And that we have a mechanism behind the phenomena, what we see. The other one is the biorhythm theory. Have you heard about biorhythm theory? Please hands up who have heard about that. So that was a quite popular theory from the 70s. As far as I know, maybe still today it can be heard about that. The principle of that roots back on a physiologist on a family doctor actually, who was a patient of Sigmund Freud in the 19th century, who liked pretty much the numbers. So it roots back in the number mysticism. The principle of this theory is that these rhythms have a start point, and this start point is the day of birth, exactly the day of the birth. The other funny part of this theory is that the period length is given as, I don't know, 28 days for one type of rhythmicity, or 31 days or 33 days. So exact numbers are given without any biological mechanism in the background. And what happens with people who come not for who come earlier, so whose the birthday is earlier, or I don't know. So then their fate is shifted, or I don't know. So there is no biological mechanism. It hasn't been proven. Of course, the believers of this theory try to make statistics as much as maybe they could show that this is a true scientific approach, but there is no biological mechanism which could describe why exactly those periodicities have been selected by the inventor of this theory. So this is what we know about biological rhythms, that it has a relatively stable period length, but the phase can be adjusted, which is completely missing in the biorhythm theory. So it has, this is very important to understand. And now we have reached the end of the first part. Maybe I have to speed up a little bit, but do you need a break? The next part, we make a break, a short break. The next part is about how the body clock works, how the physiology is regulated by the body clock. And to demonstrate that, a 1 hour lecture would not be enough in this type of setting, what I do here. There is a very good video available also on YouTube, which was done, has been done by BBC, and the top researchers of UK and many European research centers have been interviewed about not theoretical parts and not research part, but the practical parts of how the body clock runs healthy. So I think that's the best way to give you an insight of this aspect. So if you want, we make now 5 minutes break. Is good? Okay, so we start in 5 minutes.